So this is our second session for botany and how to do good in score test. So regarding this score, as you all are aware that it is not only just a way to win a lot of cash prize, scholarship, but it is also a way to analyze yourself. It is also a way to get ahead of other students with respect to NEET. And here with this exam, you will analyze yourself, you will see where you are good at, where you are weak, and it will also help you to move forward with confidence and more self-determination. So I guess that all of you must have enrolled till now for a score. If not, today with the link in the description you can enroll yourself for a score. And not only you, tell all your friends, your family to get enrolled in the score and see where you stand. If you are serious for NEET, then take score seriously. And as I have promised in the first session, that not only I will tell you to appear for a score, not only will I tell you what are the syllabus, what you should read, but I have promised you to cover some of the important topics. So here I am. So with the introduction of score, I guess that all of you will positively enroll for it. And now if you have already enrolled or you are planning to enroll, let's get into the academic part of it, where I will explain you few of the important topics. So as you can see here we are going to cover 3 important chapters for a score in one shot. So here today in this video I will make sure that all the important questions from this topic get covered in this class, and I am sure that if you cover this, you will do extremely good in score. So here as you can see, the heading is concept for better score. So here I will talk about different concepts, and these concepts will help you for better score, not only in this score test, but it will also help you to do better in and get your NEET score better. So it will help you in this score test as well as NEET. So let's get started. So now, as the thumbnail suggests, here we will cover 3 important chapters. So the first chapter we will cover will be the living world, in which we will see the concept of living and what are the important points in this chapter with respect to NCERT which can be asked here. Then the next we will cover is the cell, very important chapter, and all of you must be studying it from very long time. Then we will also have some of the reference material, and here I think one chapter is missed, and that is the cell cycle, right? So we will cover the first chapter, living cell, second chapter, cell, the unit of life, and we will cover the third chapter, cell cycle. And now, as said, we will also cover the reference material, and we will also cover the most important questions. So this session is going to be wonderful for you. You will learn a lot, and let's hope that you get the maximum benefit out of this and use this to improve your score. So let's get started. So as already told to all, what are the benefits of a score? So this all benefit by this time you all must be knowing. So just quickly, not only it will help you in better exposure, you will get a structured feedback, guided mentorship, you will also get a scholarship opportunity, and you will also get motivational guidance and confidence boosting session with our mentors. So let's move ahead. So the first chapter I am going to cover is the living world. Now living world, here the word living. So it starts with what actually is a living. So living organism has a certain character. So from here example and the questions are asked that what constitute a living organism. So living organisms show growth. Now growth is increase in size. So is only living organism grow, or non-living organism can also grow? So I will say even non-living organism can grow. Now you will say, no sir, how can non-living organism grow? So have you seen a sand dune which is growing, right? Even you all must have seen the garbage pile, it keep on growing. It is non-living, but its size keep on increasing. The Mount Everest, we say that Himalayan mountain height is increasing every year. So these all are non-living, but they are showing growth. What are these growth? These growths are called external growth. It is called external or extrinsic growth. Extrinsic growth is a characteristic of non-living, whereas the intrinsic growth means growth from inside. It is the characteristic of living organism, right? So when I am telling here growth is the characteristic of living organism, then we are talking about which kind of growth? We are talking about the intrinsic growth. Extrinsic growth is shown by non-living. So if question is asked, living organisms show intrinsic growth or extrinsic growth? So it is the intrinsic growth. Extrinsic means deposition from outside. Now we don't grow because of deposition from outside. We grow because of inner growth, right? The cell grows, the cytoplasm grows. That's how we grow. So remember this here, growth is a characteristic of living organism, but growth can also be shown by non-living organism, and that growth shown by non-living organism is called extrinsic growth, and living organisms show intrinsic growth. Then the second question is reproduction. Is reproduction done by all living organism? Can we say all living organism reproduce? The answer is no. All living organisms do not reproduce. There are many living organisms. All of you must have known about mule. Mule is the hybrid formed by cross-breeding horse and donkey. So mule cannot reproduce. It is sterile. In the same way, honeybee, the worker honeybee is also sterile, which is called drone. So there are some living organisms which do not reproduce. Although reproduction is a characteristic of living organism, but can we say all living organisms reproduce? No. There are many living organisms which don't reproduce. Then metabolism. Now metabolism means all the living reaction, all the reaction which is taking place inside us, inside any living organism, inside any cell. Sum total of all is called the metabolism. Let me give you an example. When you are eating, digestion is taking place. The nutrient is getting inside the cell and they are producing energy. So inside the cell, the breakdown of molecule, the making of molecule, plant make glucose, we break glucose. In respiration, glucose is break down into CO2 and H2O and energy is produced. So all such reactions which take place inside our body is not only inside our body but inside our cell. It is called metabolism. So metabolism include all the biochemical reaction in a body. Now it consists of two parts, anabolism and catabolism. Now anabolism, when a small molecule become bigger molecule, it is called anabolism. Small simple become large complex. For example, plant use CO2 and H2O to produce glucose. Small simple molecule CO2, large complex molecule glucose. So this is an example of anabolism. Then comes catabolism. Catabolism, reverse will happen. Here large complex molecule will break down into small simple molecule. Example, glycolysis. Here the glucose is broken down into small molecule. Digestion, large molecule broken down into small molecules. So these are example of cata. Cata means cut. Cut means large into small. It's called catabolism. Anabolism, formation of large molecule. All together it forms metabolism. Now, metabolism is shown by all living organism. Metabolism is shown by all living organism. Then cellular organization. All living organisms are made up of cell. This is the meaning of cellular organization. Can there be any living organism which is not made up of cell? No. All living organisms are always made up of cells. Then last one is consciousness. Consciousness means the respond to surrounding, right? Means I am breathing in response to the surrounding, right? My interaction with the surrounding. For example, you see plant always bend toward the light. What are they doing? They are interacting with the surrounding. Root move toward the water, interaction with the surrounding. So this is an example of consciousness. Consciousness is not just shown by animal, but it is shown by plants also, shown by bacteria also, right? Means all living organisms show consciousness, right? So now if I say these 5 points are here, now if I ask you that, here if I ask you one simple thing, that here if I say all living organisms grow, what I said? No, all living organisms do not grow. All living organisms reproduce? No. Here I said all living organisms grow, but growth is also shown by non-living organism, right? Mind it. All living organisms grow, but growth is also shown by non-living organism. Here all living organisms reproduce, but some may not reproduce. Means you will find exception in both this case, but there is no exception in these three cases. That's why they are called as the defining property. So if question is asked, what are the defining property of living organism? You will say metabolism, cellular organization and consciousness. Growth and reproduction are non-defining properties, right? So now out of this 4 you can easily say what are defining, what are non-defining. So that may be a question, that what are the or which of the following is the defining property of living organism. Clear? So this clarifies what is living. So you had made one part of the score. Now there are, again this is also taken from NCERT, these informations, but sometimes these informations become very important, not only for a score but also for NEET. Direct question is asked from this point in previous year, right? This is a previous year question where the number of species was asked. So here, diversity of living world. We know that living organisms are there. So living organism consists of large number of plant, animal, microbes, right? So this diversity, if we count, there are 1.7 to 1.8 million known species, right? Now, if it is estimated, then there are about 8.7 million species. Means, see, total it is expected that there might be 8.7 million species, but how many have we known till now? Only 1.8. Means we have not even known about 25% of the existing living organism, right? Still a lot is yet to be discovered, right? Now, when we study living organism, right, then we use 3 important things. One is called taxonomy, another is called systematics, and another is called classification. So these 3 terms are very important in this chapter. So we will see that one by one. Remember, always remember this estimated number and known number. And then you can have an idea that there are only about 25%. There are only about 25% of the species which are known till now. Now, if we move ahead, so now here as I have said, 3 terms are important: taxonomy, systematics and classification. So let's see them one by one. What is a taxonomy? Definition, direct question. identification, nomenclature and classification. So taxonomy include identification, nomenclature and classification. Along with that, another term you remember, characterization, means the basic process involved in taxonomy is characterization, identification, nomenclature and classification. Clear? Now, who is the father of taxonomy? Again, very common question. So, Linus is the father of taxonomy, right? Then further, after this is taxonomy. Then there is another second term what here we have seen, systematics. So what is systematics? Let's see, and how it is related to taxonomy. So systematics, we can simply say, in very simple term we can say systematics is equal to taxonomy plus, it include taxonomy plus evolutionary relationship. So remember this very, very important point here, that systematics is taxonomy plus evolutionary relationship. So what is the difference between taxonomy and systematics? We can simply say that if we add evolutionary relationship to taxonomy, it becomes systematics. Clear? So relationship is very important. So whenever the term systematics come, you keep it in mind, it is about evolutionary relationship. Then comes the classification. The third term was classification. So classification, for that nomenclature is important. Nomenclature is also a part of taxonomy here, nomenclature. So let's see, after that we will do classification. So first nomenclature. So here if we see nomenclature, there are many ways of nomenclature, but the most important way of nomenclature is the binomial nomenclature. Now, binomial means two words. Name will be explained in two words, right? It was also developed by Linus. So Linus is the father of taxonomy also, and Linus has given binomial nomenclature also. Now, if we see this binomial nomenclature, it is given by Carolus Linus, or Carl Linus we call him. It has two words used. First word is the genus, another word is called the species. The genus is written first, species is written next. The first letter of genus is capital and the first letter of a species is small. For example, human. Human are called Homo sapiens. Homo sapiens. So here the first word, it is genus. The second word is the species. Now the genus first letter is H, Homo. For a species the first letter is S. Now you can see clearly this one is uppercase. Uppercase is also called as capital. And this one is small letter or lowercase. Clear? So first word genus, second word species, and first letter of genus is capitals, first letter of species is small. Now if I am doing it handwritten, then I will underline them separately, and if I am typing it, it has to be written in italics. The word used here, they will be written in italics, means either you underline it, right? When you are typing it, it can be written in italics, but when you are writing it in through hand, then you have to separately underline it, not underline in one. Both the words should be separately underlined. Then these writing rules are decided by two bodies, ICBN and ICZN. ICBN will do the botanical nomenclature, that is the nomenclature of plant, and ICZN for the nomenclature of animal. And one example given, Homo sapien. For example, mango. What is the botanical name of mango? Mango is Mangifera indica. Mangifera indica. So here I will write mango. Mangifera indica. So Mangifera and indica will be separately underlined. Then let's move ahead. Now when we do the classification, so here the organism can be classified into several hierarchy, and this is called taxon. Taxon. And these taxon can be kingdom, phylum, class, order, family, genus, species. Means we will say like this. You can easily remember it like this. King, Philip, cut, open, 5, green, snakes. Right? So for here you can easily remember kingdom, phylum, class, then order, family, genus, species. So this correct order you have to remember. So how many categories are there? So total there are 7 categories. And how, in which order you will arrange? So this arrangement is called hierarchy. And this hierarchy is in this form. You will go from kingdom to phylum, class, order, family, genus and species. Clear? So this is hierarchy. Now which is the basic unit of classification? So you will say species is the basic unit of classification. Now as you go down, then the common character increases. Then the common character increases. Common character increases as you go down, and as you go up, common character decreases. Remember, in the hierarchy, if you go down, means from family to species, the number of common character increase. From species to family, the number of common character decreases. Then, so these were all the important points. So this order you have to remember. Binomial classification, nomenclature you have to remember. These 3 terms, taxonomy, systematics, nomenclature and ultimately classification. Then you have to remember, and then what is living. These are the 4 or 5 important points in this chapter which you have to keep it in mind. Nothing beyond this will be asked in this chapter. Now coming to the next interesting and important chapter, that is cell. So cell, you know that cell is the structural and functional unit of life. Now a unicellular cell organism can be unicellular or they can be multicellular. A unicellular organism can live independently, whereas multicellular organism, different cells will perform different function. In our body, we are multicellular. We have different organ. Heart is performing its own function. Lung is performing its own function. Right? So liver is performing its own function. So different organ performing different function. Multicellular. In unicellular bacteria, same cell is doing digestion also, respiration also. Every process is done by the same cell. That's why they live independently. Who discovered cell? So cell was discovered by Leeuwenhoek. Leeuwenhoek discovered the living cell, and Robert Hooke discovered the dead cell. So first cell was discovered by Robert Hooke, but he discovered dead cell. Now nucleus was discovered by Robert Brown. Robert Brown discovered the nucleus. Cell theory, very important point. Now all the information of cells were kept together and a cell theory was made. The cell theory was given by Sladen and Schwann. So Sladen and Schwann has given the cell theory, but who completed the cell theory? So it was Rudolf Virchow. So credit for complete cell theory goes to all the three scientists. And what they have said? One, the Sladen said that all plants are made up of cell. Now, so Sladen was a botanist, so he worked on plant. Now, Schwann was a zoologist, so he worked on animal, but he also worked on plant. So he said that the animal cell have a plasma membrane and plant cell have a cell wall. Overall, they have given that all animal plant are made up of cell, but they were not able to explain how the cell is originating. So this was said by Rudolf Virchow, and he said that all cell arise from pre-existing cell. So Rudolf Virchow has said about the origin of cell, and this completed the cell theory, and this is the modern cell theory. All living organisms are made up of cell and their product, and all cell came from pre-existing cell. So this is cell theory. If we do the overview of the cell, so here we know that the cell consists of the plant cell, animal cell. So if I see the overview of the cell, so plant cell will have cell wall as well as cell membrane. Animal cell only have cell membrane, no cell wall is present in the animal cell. Inside the cell, nucleus, if it is a eukaryotic cell, then the nucleus present, which is membrane bound, and it consists of chromosome, which is made up of DNA. So cell has nucleus, and inside the nucleus is chromosome, which is made up of DNA. Inside the membrane, the fluid matrix is called the cytoplasm. We call it semi-fluid matrix, which is the arena of cellular activity or site of cellular activity. So very important component of cells are cell wall, cell membrane, nucleus, cytoplasm. Cell wall only present in plant, or you can rather say cell wall is present in plant and other organism also, like bacteria. We will see it in a while, but we can clearly say that animal cell totally lacks cell wall. Nucleus present inside the cell, but only in the eukaryotic cell. And then cytoplasm, it is the site of cellular activity. Now there are two types of cell, prokaryotic cell and eukaryotic cell. Prokaryotic cell, there is no membrane bound nucleus. Membrane is absent in nucleus. So this we can say that it is not a true nucleus. So we call it, instead of nucleus, we call it nucleoid. Nucleoid is present in the prokaryotic cell. In eukaryotic cell, here nucleus is also present and membrane bound cell organelle is also present, which is absent here. In prokaryote, no membrane bound nucleus or other cell organelle, whereas here both are present. Now if we talk about the cell organelle can be of three types: membrane bound cell organelle, non-membrane bound cell organelle. Now three type, why I said? Because this membrane bound can also be divided into two parts. One is the single membrane bound and another is the double membrane bound. So in this membrane bound, if I put a red circle, that is double membrane bound. So for example, here mitochondria. Mitochondria is double membrane bound cell organelle, and if we consider plant cell, then plastid. which include chromoplast, chloroplast and leucoplast. That is also a double membrane bound cell organelle. Then here if we say nucleus is also double membrane. So remember, 3 double membrane bound cell organelle: nucleus, then mitochondria, and then plastid or the chloroplast. Non-membrane bound cell organelle: ribosome, centrosome. So question can be asked, which of the following is double membrane bound, single membrane bound, without membrane bound cell organelle? So you can easily answer it. Then if we have just seen that there are 2 types of cell, prokaryotic cell and eukaryotic cell. So what are the characteristic of prokaryotic cell? So the characteristic are, prokaryotic cell includes bacteria, cyanobacteria and mycoplasma. They all are prokaryotic. Now already I have told you the characteristic, that they do not have membrane bound cell organelle. Membrane bound cell organelles are absent. Now if I further say here that their shape, so they have a very simple shape. They have a very simple shape present. They have a very simple shape present. They have 4 type of shape. Bacillus means rod shape. Then they have coccus, that is spherical. So here I will draw and I will explain. So this is the bacillus, means rod shape. Then another one is spherical, that is coccus. Then there is spirillum, right? This is spirillum. And the last one is comma shaped, like this. So this comma shaped is called vibrio. So these are the 4 shape: rod shape, spherical or circular coccus, then spiral and vibrio. Now the DNA in prokaryote will be circular, and since nucleus membrane is absent, so no membrane is present around the DNA, means it is talking about nucleus. Ribosome is 70S. There are 2 types of ribosome, 80S and 70S. 70S is the smaller one, 80S is present in eukaryotes only, and this is the 70S. Inclusion include gas vacuole, glycogen, etc. What are inclusion? These are the storage bodies. Then they have mesosome, they have plasmid, and they have cell envelope, which is 3-layered. Now 3-layered cell envelope means what? So we have just seen cell wall and cell membrane. These are 2 layers of cell envelope. The 3rd layer is the capsule. So if I write it like this, outermost layer is called capsule, or sometimes we also call it glycocalyx. Then the inner layer is the cell wall, and then inside that is the cell membrane. So 3-layered cell envelope means the 3-layer is capsule, cell wall and cell membrane. All 3 together are called cell envelope. Now remember, capsule or glycocalyx is only present in bacteria or prokaryote. They are not present in eukaryote. Plasmid, so plasmid is circular DNA. Plasmid is the circular DNA which is present in the cytoplasm. It is not a part of nucleoid, it is present in the cytoplasm. Mesosome, it is the infolding of the membrane. If I right here. Here if I mention mesosome, so this is the cell. Now the infolding like this. This infolding in the membrane, this is called the mesosome. Remember, the mesosome is the infolding of the membranes. So understood here, mesosome, plasmid. Plasmid is randomly present as a circular DNA, and then the cell envelope, right? You have 3-layered envelope present in the bacteria. The outermost is the capsule, cell wall, and cell membrane. Then on the basis of cell wall, bacteria can be divided into gram positive and gram negative. See, let's say you have a white shirt and you color it yellow. You put it in yellow color, it will become yellow. Now you put it again in white color. In normal water you put. First you stain it in yellow color, then take it out, put it in normal water. When you take out, if the yellow color is still in the shirt, means shirt has retained the stain. If yellow color is gone, means shirt has lost the stain. Lose the stain means negative, and retain the stain means positive. In the same way, bacteria also have certain character that the cell wall retain the color. If it retain the color, if it retain the stain, it is positive. It is lose the stain, so it is negative. So bacteria is divided into gram positive, gram negative. Positive if it retain the stain, negative if it lose the stain. And you can easily see under the microscope, one is stained, colored, blue or purple in color they will appear, other will appear colorless. So that will show the staining. Then comes the motility. Now motility is through flagella, pili and fimbriae. So 3 organ of, so in this structure they will have a very long flagella, and the smaller one they are called pili and fimbriae are present. So pili, fimbriae and flagella both are present on the surface of the prokaryote. So this is all about the prokaryotic organism. So from the shape to the envelope, then to the mesosome, plasmid, inclusion body, ribosome, motility, these all are important points which you have to cover. Then coming to the eukaryotic cell. Eukaryotic cell can be plant and animal. So if we say plant cell will have cell wall, which is absent in animal cell. Plastids are found in plant cell, whereas they are absent in animal cell. Vacuoles are large in plant cell, small or absent in animal cell, and centrioles are present in animal cell but they are absent in plant cell. So this is basic comparison between plant cell and animal cell, which most of you might be already knowing, but important, so question can be asked from here. Then coming to the individual cell organelle. If I say cell membrane, so cell membrane is double membranous structure, right? It is, we call it phospholipid bilayer. Cell membrane is phospholipid bilayer. It is made up of lipid as well as protein. So we can say it is lipoprotein. It is made up of lipid and protein. Who discovered this? So fluid mosaic model was given and Singer and Nicholson were the name of the scientist. He said that Phospholipid is bilayer and it is made up of lipid and protein. If there is membrane, so there can be transport across membrane, and transport can be passive or active. Active transport need ATP, passive transport no ATP is required. So this is the cell membrane. Then cell wall, which may be present in some organism, so it is present in cell wall, it is present in bacteria, but if we say it is present in plant cell, so plant cell wall is made up of cellulose. In fungi it is made up of chitin, right? Now remember this, cell wall is not always made up of cellulose. Only plant cell wall is made up of cellulose. Fungal cell wall is made up of chitin, and bacterial cell wall is made up of peptidoglycan. Peptidoglycan. This is for bacterial cell wall. Is it clear? Now, cell wall can have different layers: primary, secondary and middle lamella. So these are the three layers of cell wall, and this cell wall may have a small passage, right? It may have a small passage. It is called plasmodesmata. Remember, it is a passage in the cell wall which is called plasmodesmata. Then further, if we see, there is a system of membrane which is called endomembrane system. It consists of endoplasmic reticulum, which has a function of protein synthesis and lipid synthesis. Rough endoplasmic reticulum, RER means rough. Rough endoplasmic reticulum, protein synthesis. S for smooth. Smooth endoplasmic reticulum, lipid synthesis. Then Golgi body. So Golgi body modify and pack protein and lipid. So packaging and modification is the work of Golgi. Then lysosome, you all know, it is also called as the suicidal bag of the cell, lysosome. So lysosome has hydrolytic enzyme and it is involved in digestion, and because of this it is called the suicidal bag of the cell. Suicidal bag of the cell. The vacuoles are present inside, and vacuoles are the store. They are used for storage, and they also provide pressure into the cell. They maintain the pressure inside the cell. Cell do not become very loose. It remain turgid. After that is the mitochondria. You all know mitochondria is called the powerhouse of the cell. Why it is called powerhouse? Because it produces ATP. Its main function is ATP production. This mitochondria can itself divide through fission, and one become two. So they divide through fission, and they are actually double membrane structure. Outer membrane and inner membrane is folded like this. So can we say in mitochondria the outer membrane is regular, inner membrane is folded, right? Then inside is the matrix. So this mitochondria is called the powerhouse of the cell. Now plastid. Plastid include of three type: chloroplast, chromoplast and leucoplast. Their function: chloroplast help in photosynthesis, chromoplast help in, it has pigment, so it will give color, different color other than green. Leucoplast, storage, right? Its function is a storage. Now inside the chloroplast there is grana and there is stroma. This is also double membranous structure. Outer membrane, inner membrane, and inside is this stacked structure. This stacked structure is called grana, and this part is called the stroma. Grana has unit called thylakoid, right? So these both are double membranous structure, this and mitochondria, and their matrix was present, here it is stroma present. Then comes the ribosome. As I said, it is of two type, so one, and each ribosome has two subunit, right? Each ribosome has two subunit. Now there are two types of ribosome, one is called the 70S, another is called the 80S. 70S has two subunit, larger one is 50S, smaller one is 30S. 80S, larger one is 60S, smaller one is 40S, right? So these are the subunits which are present. We know that 70S is present in prokaryotes and 80S in eukaryotes. Now remember one more thing, in eukaryotes the prokaryote, this 70S is also present in mitochondria and chloroplast, remember, which are part of eukaryotic cell. So chloroplast and mitochondria both can have 70S ribosome. So in short, can we say that eukaryote will have both 80S and 70S, but prokaryote will have only 70S. That is the clear difference. Then the cytoskeleton, so they will provide shape, movement and transport. They will help, so this is the function, and it is of three type: microtubule, microfilament and intermediate filament. Cilia and flagella, it is present on the outer part of the cell. Longer one is called flagella and the smaller one is called the cilia, right? So one help in attachment and movement, another help only in the movement. Cilia is short and it will be multiple, as you can see here, cilia, whereas flagella will be long and it will be less in number. The structure of this cilia and flagella is said to be 9+2 arrangement, and they also have a basal body. See this base part, this part is called exoneme and this part is called basal body, right? This exoneme has 9+2 arrangement, right? And then there is a basal body also. Then comes the centrosome and centriole. So there are centrosome, each centrosome has two centriole. It is like this. Two centrioles and they are perpendicular to each other, right? So this is one centrosome which has two centriole and they are perpendicular to each other. And these are having this, here it is 9+2, they are called 9+0 arrangement. So centriole has 9+0 arrangement, whereas the exoneme of flagella and cilia has 9+2 arrangement. Their function, so they will help in spindle formation and cell division. Spindle formation and cell division. Clear? So this is again very, very important. Then comes the nucleus, one of the most important part of the cell. It is basically the controlling part of the cell, brain of the cell, or just like in computer, the processor of the computer, it is the processor. So it consists of envelope called nuclear envelope. It is two-layered, right? It is two-layered. Then inside the nucleus there is a nucleoplasm, a dense structure, spherical, right, like this, and it is without membrane. Without membrane. Then chromatin is the DNA, thread-like structure, DNA. So chromatin is made up of DNA, although some RNA is also there, some protein is also there, right? And this is the nucleolus, which, so this is actually the nucleolus, right? Inside the nucleoplasm is the nucleolus. Basically nucleoplasm will have all this together, and inside is the dense structure which is called the nucleolus, which is present without the membrane. Then the chromosome type. Chromosome can be metacentric, now let's first understand the structure of chromosome and then I will explain. Let's say this is the chromosome. So the central part is called the centromere and this one is the chromatids, right? Now if this centromere is at the center, then it is called metacentric. Now if the centromere is away from the center like this, it is called submetacentric. Now if it is totally at the end like this, toward the end, it is called this one acrocentric. And if it is totally at the end like this, if it is at the end, it is called telocentric. NCERT has example of all the three. And the last one, microbodies. So microbodies include peroxisome and glyoxysome. Peroxisome, so they are very small membrane bound structure which is present in eukaryotic cell. Peroxisome, it has a role in detoxification. In glyoxysome, it has a role in conversion of fat to sugar. So these are some of the important things which you have to keep in mind while dealing through the cell. Clear? So I guess that you all might have understood this part. So this is giving you an overall idea, right? If keeping these idea in mind, if you read NCERT, then every point will get covered, right? And rest assured, all the question in a score, nothing will cross this. There will be nothing which I am discussing here which will be there in the score. Then the last part is this. This is the cell cycle. Now you can clearly see that cell cycle is, now cell is dividing, right? In simple term, if we see, then cell cycle is divided into two phases. One is the interphase and another is called the M phase. You can see here, see, if this is the entire cell cycle, this part is the interphase and this part is the M phase. This is your M phase and this part is the interphase. You can clearly see that interphase is longer, M phase is shorter, right? Now this interphase is divided further into G1 phase, gap phase 1, S phase and G2 phase. Then the M phase is divided further into karyokinesis and cytokinesis. Karyokinesis and cytokinesis. Here the karyokinesis will be further divided into prophase, metaphase, anaphase and telophase, right? Prophase, metaphase, anaphase, telophase and finally the cytokinesis. To all together this form the cell cycle. As you can see here, G1 is the largest phase. Here protein, right, RNA and cell organelles are synthesized. Cell organelles are synthesized. Here DNA synthesis take place. Here centrosome duplication take place. Here again protein and RNA synthesis take place. And then followed by prophase, metaphase, anaphase, telophase and cytokinesis, which all together consist of the M phase, right? Now this is a cell cycle showing the mitosis, right? You can see mitosis. Now the karyokinesis will be two types in case of meiosis, right? Cell if continue to divide, then it will not enter into G1 phase, and if cell has to divide, it will be present, means it stop cell division. When cell has to rest, then it will get into the G0 phase, and when it again want to divide, it will get into the G1 phase, and it will continue the cell cycle. Now this cell cycle is of two types. This is called the mitosis. Several stages of mitosis: prophase, metaphase, anaphase, telophase. The cell divide into two, and this is the meiosis, in which one cell dividing twice, and in the end we are getting 4 cells, right? So in mitosis we are getting 2 cells, in meiosis we are getting 4 cells. Now in meiosis there are 2 nuclear division taking place, means 1 synthesis and 2 division. That's why it is getting 4 cells. Here we start with 2n and we get a cell which is n, right? So in meiosis the chromosome reduced by half. Reduced by half, right? So now here what you have to think, that one very important thing which happen here is the crossing over. So two important characteristic of crossing over is, the first one, chromosome number become half, and second is crossing over, which is a source of variation which lead to evolution, and another is the number of chromosome remain throughout the generation, it remain the same because the cell number become half, right? See, this is a little contrasting. If you see that in order to keep the number constant throughout the generation, the chromosome number is reduced by half. For example, human is having 46 chromosome, gamete will be 23, then only next generation will be 23 plus 23, 46, and in this way throughout the generation the number of chromosome is maintained, right? So these are the some simple basic difference between mitosis and meiosis, right? So as you can see, in meiosis there is crossing over, in mitosis no crossing over. In mitosis pairing of chromosome take place, in meiosis pairing of chromosome take place, mitosis no pairing of chromosome take place. Here 2 division is taking place, here only 1 division, here 4 daughter cells are formed, here only 2 daughter cells are formed. Here the daughter cells forms are n, means the number of chromosome become half, here the number of chromosome remain the same. So these are some of the important points. Now what you have to do, you have to go and solve the exemplar question like this. All these are exemplar question from the NCERT, right? As you can see here, these are the exemplar question. Now one question, if I say, let's say this, because just we have covered meiosis. So meiosis occur in organism during, so when it occur during sexual reproduction, right? It do not take place during vegetative reproduction, only it take place during sexual reproduction, and that too precisely in gamete formation. In this way, if you solve all the question from the exemplar question, then it will be very easy for you to do good in score. So now with this session, in this session I have explained you what to read in these 3 sections. Some of the concepts also I have explained. Now if you go back, start reading NCERT, read it topic by topic, then everything will get concrete. And you should share this so that your other friends also get idea about what to read in the score. And as I said, only NCERT you can refer, that is more than sufficient for your score. Already you are aware about the syllabus. If not, in the descriptions, the syllabus, pattern of exam, everything is given, or you can go back in my first session where I have explained all the things related to pattern. I have also explained what are the most important 25 topics. That video also you can see. So finally, if you like it, just like and share with your friends, and don't forget that the score exam is on 28th September and 12th October. I know that most of you by now must have registered for it. If not, link is there in the description. Go and quickly register it. Also tell your friend, your neighbor, your relative to register for NEET score, because one decision can change your life. And thank you all of you for being with me in this session. Next week another bio session will be there, and I will meet you after that week. So till then, take care and keep scoring.